As a kid, have you ever dreamt of going on long space missions or envied the astronauts who did? Or perhaps envisioned living on another planet? Well, unfortunately, your dreams were crushed when you turned about 10, and your parents told you about how unsafe it was. But here comes the question, why is it unsafe? One of the main factors is radiation. But what does it do to people? According to studies, cosmic radiation can penetrate bodily tissues, leading to DNA damage, increased cancer risk, and potential harm to vital organs. Moreover, prolonged exposure may also impair cognitive functions, accelerate degenerative diseases, and increase the likelihood of cardiovascular issues. The effects are cumulative, making it hazardous for astronauts on deep space missions and pose problems to potential human life on other planets.
But how do we truly understand the full impact of cosmic radiation? That's where our experiment comes in. We're exposing different microorganisms, E. coli, yeast, and the radiation-resistant Deinococcus radiodurans to cosmic radiation over various periods of time. By studying the rate at which these tiny organisms develop new traits and mutations, we can get a clearer picture of how radiation affects living cells. First, we start by growing cultures of these microorganisms in optimal conditions here on Earth to ensure that they are healthy and ready for testing. We've set up 3 groups. One will be our Earth control group, kept in Earth conditions. This setup takes place inside a thermoscientific formal environmental chamber with a controlled temperature between 22 to 25 degrees Celsius and maintains an oxygen level of 21%. This chamber is connected to a Sanyo MCO18AC carbon dioxide incubator, which ensures that the CO2 level remains at 0.04%, perfectly mirroring Earth's atmospheric composition. This creates a reliable baseline for comparison to ensure that any observed effects are accurately attributed to the experimental conditions rather than to natural variability or external factors. The third, our experimental group, will be directly exposed to cosmic radiation. The latter two will take place in the Deep Space Gateway, located outside Earth's magnetosphere. It provides a unique opportunity to observe how these microorganisms respond to prolonged radiation exposure in microgravity.
Next, the microorganisms are carefully transported to the Deep Space Gateway (DSG) aboard the SpaceX Dragon spacecraft. The journey takes approximately 6 days, during which the microorganisms are maintained in carefully controlled conditions. Temperatures inside the transport containers are kept at a freezing -80 degrees Celsius, while the containers themselves are engineered to withstand the drastic pressure changes from Earth's sea level to the vacuum of space. Humidity levels are regulated between 30 and 60%, ensuring the integrity of the samples, while specialized ventilation systems actively manage carbon dioxide and oxygen levels. The containers are insulated using closed-cell polyethylene foam to maintain temperature stability and absorb any shocks during the journey. In the microgravity environment of space, Velcro straps are used to secure the containers, preventing them from floating freely. Once they've reached the DSG, they will face unfiltered radiation levels ranging from 1.4 to 1.9 millisieverts per day, found outside Earth's protective atmosphere. To study these effects over time, the experiment will run for 3 distinct exposure periods: short-term for 1 month, medium-term for 6 months, and long-term for a full year. Throughout the experiment, critical conditions like temperature, atmospheric pressure, humidity, and gravity will be carefully controlled. Additionally, we will use a chemostat to ensure that the microorganisms remain in their optimal growth phase, allowing us to continuously monitor their biological processes under these extreme space conditions. We will also monitor the growth and activity of these microorganisms in real time, using advanced sensors and communication systems to send data back to Earth. Once the exposure period is over, the microorganisms will be returned to Earth for detailed analysis. Using high-throughput sequencing and bioinformatics tools, we'll compare the genomes of the exposed microorganisms with those from the control groups to identify any new traits, mutations, or genomic alterations. Is a Python file which will be used for transmission. In a DNA, there are 4 types of data: A. adenine, T. thymine, C. cytosine, and G. guanine. All of these chemicals are arranged in a particular order to make the DNA. To measure genomic mutations, we look for changes in the order. Data here will be extracted from Biotech H1 Hybrid Multimode Reader. However, as we do not have access to this device, we will be coming up with a sequence using OpenAI ChatGPT. First, we will start by creating the variables. DNA start, which is in this case equals to 5, DNA end, which is equal to 3, and a sequence, which is a string made up of A, T, C, and G. 5' refers to the start of the DNA strand, where the phosphate group is attached to the 5th carbon atom of the sugar. 3' refers to the end, where the hydroxyl group is attached to the 3rd carbon atom of the sugar. A, T, C, and G are the various chemicals making up the DNA. Next, we will split this sequence of DNA into letters. This variable called DNA sequence holds a list of letters in this sequence. Next, we will convert this to amplitude for transmission of sound by radio. The start of the amplitude will be 20 plus the DNA start, and the end of the amplitude will be 20 plus DNA end. This also converts the sequence into amplitudes. Finally, it is all represented by this list called wave. These values are in decibels and are converted to a wave using the software Audacity to create the sound to be transmitted by radio. The rules are A, T, C, and G have the amplitudes of 11, 12, 13, and 14 respectively. The starts and ends have the amplitude of 20 plus their own number. The final transmission wave can be seen here. Next, we will receive and decode the sound. For simplicity's sake, we will make the received value equal to the wave here. The start of the received wave will be the first number of the wave.